Okay, thank you very much. Thank you, Ivan. Thank you, Massimiliano, for inviting me. Okay, I will. So I will be presenting quickly what we have been obtaining since we working together since 2003, and the group is a kind of a multidisciplinary group composed of Diego and Rodolfo of us physicists, Greta and I are statisticians, and Alberto is a molecular biologist, and Ellen and Lutz German mathematicians. So basically we study the mathematical structure of the genetic information through the paradigm of information and communication theory. We have two interconnected projects. The first one concerns a mathematical model for the genetic code based upon number theory. I will try to skip off the mathematical details and focus on the practical and biological implications. And a second one I'm skipping is about circular codes and translation. So yes. So as you know, focusing on the first one, as you know, the genetic code is a mapping, is a degenerate mapping, and it has a degeneracy distribution. Degeneracy means that, for instance, we have two amino acids, isoleucine and cysteine. These two guys that have degeneracy 3 means that they are mapped. They are represented by three codons each. So the question is there a mathematical representation of the mapping? And can we use number theory to achieve the task? So this is the question that we were faced in 2003. And we can describe the degeneracy distribution of the genetic code through number theory. But in particular, representation system of integer numbers. But, you know, representation system, usually the numbers we all have learned are power-based, meaning that, for instance, if we think in the binary system and we use the power of the base 2 to represent numbers. For instance, here, number 17 is represented by this binary string univocally. So how can we make this system redundant? Because this is of no use to represent a redundant mapping like the genetic code, where we have 64 codons and 20 amino acids plus start and stop signals. One way of achieving this is to replace this power of this sequence of the power of the base 2 with something that grows slower. For instance, let's plug in the Fibonacci, the first 6 Fibonacci numbers here. If we plug in something that grows lower, like the Fibonacci numbers, you see, these two strings both represent 17. So we have a redundant representation system. And the question is there a redundant representation system that describes the degeneracy distribution of the actual genetic code? The answer is yes, and the solution is unique. So this is the solution, and you can see in this case this is the genetic code. We have number 16 represents isoleucine and number 7 represents cysteine, and these 64 strings are mapped onto the 64 codons. So is this a mathematical coincidence? We think not, because then we started studying the mathematical properties of this model, and we think that a good model is a model that says something and predicts something that can be eventually or possibly experimentally verified. And so by matching, so this model matches all the known symmetries of the genetic code, first of all, and allowed us also to discover new symmetries that were previously uncovered. So that's a good hint. And so let's put the model all together. So you have the 64 strings and the numbers. But for instance, let me just mention, okay, what can we do of this? For instance, let's think about the parity of the strings. The parity of the strings is a purely mathematical quantity derived from the strings here. Surprisingly, the parity of the strings can be described in terms of the biochemical properties of the codons. In particular, there are the last two bases of the codons. So you can devise an algorithm that asks binary questions for the chemical classes of these two bases of the codon. And what you get, so this biochemical algorithm corresponds exactly to the parity implied by the model. And this we called parity, we call dichotomic class, and there are 3 dichotomic classes. So what did we do with these dichotomic classes? For instance, we proved that these are nonlinear functionals. So what's the meaning of nonlinear functions? It's a new pair of glasses to look at genetic information. And they are linked to a group framework. So are they good for something? Yes. We were able to, by looking at the binary sequences of dichotomic classes, we were able to pinpoint, to uncover new stuff, new information, as Chris Adami pointed out. The information is there, but it's encrypted. And this is a new way of looking at the information, because we have nonlinear functionals. And so I'll make it brief. So the dichotomic classes uncover the presence of universal short-range correlations. Moreover, interestingly, we found out that coding sequences are entropy optimized with respect to dichotomic classes. And this is really similar to what happens in efficient communication systems that require correlation optimized sequences. For instance, those described by this book by Golomb, who was one, I'm sure engineers know Golomb. That's it. So I'm skipping this. Let's focus on this. Second connected topic is circular codes and translations. So in the focus is the reading frame maintenance. That is the ability of the ribosome to read the messenger RNA without incurring in frame shifting errors. Of course, needless to say that it's of primary importance, but to date, the mechanisms by which the correct reading frames maintained remains unclear. In 1957, we all know that Crick hypothesized the existence of a comma-free code, which is a self-synchronizable code. And we all know that, unfortunately, his conjecture was proven wrong. But some 40 years later, in 1996, two French bioinformaticians just analyzed the codon usage in the 3 reading frames of a large set of sequences and found out that the most recurrent codons, they identified 20 most recurrent codons for each frame. And they found out that these 20 formed a sort of code which they called circular code because it's a weaker version of comma-free codes. They are a weaker version and they allow to retrieve the frame. They are allowed to detect the right frame. So we started working on that since 2010, and our main results, first of all, we studied a particular set of codes with the desirable mathematical properties, which I will skip. But just let me just mention that we laid the theoretical foundation by using group theory. We laid the theoretical foundation mathematically, of circular codes under the idea of group theory. And You may ask whether this is just a mathematical divertissement or not. We think it's not, because besides this theorem, we were able to connect these theoretical results with experimental evidence by using the notion of code usage. So the idea is to pass from the codon usage to the code usage. The codon usage is just the usage of a single codon. The code usage is just the usage of 20 of a code, so the 20 codons together. By using the notion of code usage, we were able to allow universal structures behind the heterogeneity of the codon usage. And these universal in that they do not depend upon species, stacks, and so on. Moreover, they are found in the 3D frames. They just hold for codon sequences and not in introns. But most importantly, and this is a working paper that it's under revision, the code coverage is strongly correlated with translation efficiency, which is experimentally measured. So we were able to establish a connection between the theorem and experimental evidence. That's it. Let me just mention that in 2006, Chi-Chi Mei edited this nice theme issue in EEE Engineering and Biology and Medicine. And 10 years after, if you're interested, we edited a theme issue of Philosophical Transaction of the Royal Society, A. And the title is similar. So if you're interested, please let me know. And last but not least, we are looking for partners interested to cooperate with us because we are establishing a cost project. Funding organization, the cost is a funding organization funding by the European Union, but it's not just restricted to European partners, so everyone is welcome. And the idea is to link information codes and function in biology. So we will really welcome everyone, especially experimentalists. Thank you very much.
Time for a question as we switch over. So, the series that you showed for, instead of the power series, the one that you uncovered for the code, if you were, for instance, to expand the code into a fourth nucleotide,
yes,
would that give you some predictive power on how you should, or?
What do you mean? To have a code with the tetranucleotides?
Yeah.
We have yes, we have we have works on that. We have work on that we can discuss. But we have I haven't shown that we can devise a model for the degeneracy, for the how the degeneracy is produced by using what we call a tetracode, which is based a genetic code based on tetranucleotides and a set of adapters, ancient tRNAs, and so again we have a paper on that in the Royal Society Interface Focus. We can discuss if you want. Thank you.
Thanks.